Hello friends, again welcome to my channel. Now today we are focusing on one of the important topic from the genetics, that is the prokaryotic transcription process. Now within this prokaryotic transcription process, we know that the central dogma for the protein synthesis, there is the synthesis of messenger RNA from the DNA, and from the mRNA then there is the synthesis of polypeptide chain, that is the protein molecule it will be synthesized. It is considered to be the central dogma for the protein synthesis. Means within the protein synthesis the initial step, it is the prokaryotic transcription process. So today we are focusing on how this process of the prokaryotic transcription it can carry out within the prokaryotic cell. Okay. Now preferentially this transcription process, again it is an polymerization reaction like that of the DNA polymerization during the DNA replication process. Similarly, this transcription process it is also a polymerization reaction, and this polymerization reaction it is catalyzed by a single enzyme which is a multi-subunit enzyme, that is the RNA polymerase enzyme. This RNA polymerase enzyme it is differentiated from the DNA polymerase enzyme in the different ways. The one of the most differentiating characteristics it can directly initiate the RNA synthesis. Means it is not possible. This de novo synthesis of the this de novo synthesis characteristics it is not present within the DNA polymerase enzyme, because it will require a presence of free 3' hydroxyl group to which it can add the nucleotides. Means the pre-existing chain it can elongated by the activity of DNA polymerase enzyme, but such type of the requirement these are not essential for the activity of RNA polymerase enzyme. Here there is the requirement of ribonucleotides, then again there is the requirement of single stranded DNA as an template. The RNA polymerase enzyme, or the one of the important characteristics of RNA polymerase enzyme, it has its helicase activity. Means a specific helicase enzyme it is not required to obtain a template strand for the RNA synthesis. It has the ability of unwinding and the rewinding of the DNA strand, that is the template DNA strand. Now preferentially this process of the transcription it can be differentiated into the 4 different stages. That is the first initial step, it is the recognition and binding of this RNA polymerase enzyme to the promoter sequences, that is the initiation site for the transcription. The secondly there is the process of initiation of transcription, then the elongation, and the third one it is the termination and release of the mRNA chain. Now we are switch over to the structure of the main polymerizing enzyme within this transcription process, that is the RNA polymerase enzyme. Now this RNA polymerase enzyme it is an largest enzyme and made up of 5 different subunit. That is there is the presence of 2 alpha subunit, 1 beta subunit, another beta prime subunit, and the sigma factor. These are the 5 different subunit. Whenever there is the presence of all of these 5 subunits within the RNA polymerase enzyme, it is considered to be the holo enzyme, and the molecular weight of this it is the 4049 kilodalton. Now the 2 alpha subunits which are present within the RNA polymerase enzyme, now these are having the ability to hold all of the subunit of the enzymes together. The beta subunit it is responsible for binding or linking the nucleotides together during the polymerization reaction, and the beta prime it has ability to binds with the template strand. Then the sigma factor, this sigma factor it is required for the initiation of transcription process, that is it is required for the promoter recognition. And whenever there is the combination of core enzyme, that is the 2 alpha subunit, 1 beta and beta prime with the sigma factor, then it is considered to be the holo enzyme. Now let us start with the how there is the initiation of transcription process. Now this process of the transcription it can be starts at the promoter sequences and which is end at the termination signal. Mostly or majorly compulsory this process of the transcription it only proceeds within the 5' to 3' direction. It forms a temporary RNA DNA hybrid. Means during the initiation process there is the formation of RNA DNA hybrid whenever there is the binding of this mRNA with the DNA strand. It is called as RNA DNA hybrid, and it has a complete processivity. Continuously there is the synthesis of RNA. It can be carried out by the activity of RNA polymerase enzyme. Now here we are switch over to the what is the promoter sequence, that is the structure of initiation sequences from which there is the actual process of transcription it can be initiated. So these are called as the promoter sequences. Now within these prokaryotic promoter sequences, majorly the two specific structures these are present, that is the -10 base pair sequences and -35 consensus sequences. This -35 consensus sequences, or within both of these, this minus sign, it indicates majorly the nucleotide sequences which are present before the transcription initiation site. Due to that, these are indicated by the negative sign, as these are present before the transcription initiation site. Okay. This -35 consensus sequence, it is the TTGACA, and it is the strongest sequences present within the promoter. After that, the -10 base pair sequences, which are the AT rich sequences, that is this sequence, it is the 6 base pair sequence, TATAAT sequence. 6 base pair sequence it is present, and majorly these are recognized by the sigma factor, which is the one of the subunit from the RNA polymerase enzyme. Now majorly the RNA polymerase enzyme, it starts the transcription process at a promoter sequence and it ends at the termination signal. It proceeds within 5' to 3' direction. Now the sigma factor, which is one of the important component within the RNA polymerase enzyme, it is essential for the recognition of promoter sequences. Again, it is responsible for stimulating the process of initiation of transcription process. It combines with the core enzyme, due to which the RNA polymerase it converts into the holoenzyme, which pauses the open hand conformation, and due to that it is fixed onto the DNA template, but it is not responsible for stimulating the elongation process. Means no doubt this sigma factor, it is essential for the initiation of transcription process, but there is no requirement of this sigma factor during the elongation process. So the elongation process of transcription, it is stimulated by only the core enzyme, because there is this sigma factor, it is get fall off after the 4 to 9 nucleotides whenever these are incorporated within the mRNA chain. Now the different variants of these sigma factors, these are associated with the RNA polymerase enzyme. Means within the different organism, different types of the sigma factors, these are present. Even only within a single E. coli cell, there is the appearance of 7 different types of the sigma factor. Because majorly these sigma factors, whenever there is any type of the variation, it is observed during the promoter sequences. The different sigma factors, these are responsible for recognizing the different types of the protein, and whenever there is the appearance of the adverse conditions within the during the growth period of these microbial cells, at that time a specific type of the enzymes or proteins, these are get expressed. So for the synthesis of this specific type of the enzymes or proteins, then there is the activation of such type of the sigma factors. This sigma factor, these are showing the variability. These are much less conserved than the other RNA polymerase subunit. Means these are not conserved one, because there is the variation within the sigma factor. The E. coli has the 7 sigma factor. Then the Bacillus subtilis, it possesses the 10 different types of the sigma factors. And majorly these different sigma factors, these are respond to the different promoters. Means for the synthesis of heat shock proteins, again the different types of the sigma factors, these are present. Here there are the sigma variability within the E. coli cells. The common form of the sigma factor, it is the sigma 70, which are responsible for recognizing the common promoter sequences that we are discussed earlier, that is the -35 consensus sequence, that is TTGACA, and the -10 TATAAT sequence. It is an housekeeping sigma factor, means it is constantly active sigma factor whenever there is the process of transcription, and other type of the sigma factors, only in the specific conditions there is the activation of such type of the sigma factor. Another example, it is the sigma 54. it has the sequence -35 sequence CTGGCAC and the -10 TTGCA. It is an alternative sigma factor involved in transcribing the nitrogen regulator genes. It means within the nitrogen fixing bacteria, majorly there is the presence of such type of, or whenever there is the content of nitrogen, it is variable within the nutrient media. At that time there is the expression of this sigma 54 and such type of the proteins, these are get synthesized within the cells. Then next one, it is the sigma 32. It is an heat shock factor involved in the activation of genes after the heat shock. So such type of the variability it is observed within the sigma factor within the E. coli cells. Now we are switch over to the main steps within the transcription initiation process. Now within the transcription initiation process, that is the recognition and bind polymerase enzyme, it is carried out with the help of the sigma factor. So firstly, within the initiation step, there is the formation of closed promoter complex. Then after there is the formation of open promoter complex. Then the ternary complex, that is involves the RNA, DNA and the enzyme. It includes the abortive initiation. Means whenever there is the binding of firstly the RNA polymerase enzyme with the promoter sequences and there is no melting of the DNA strand, means whenever the RNA polymerase enzyme it binds with the DNA double helix, then it is said to be the formation of closed promoter complex. Whenever there is the melting of the DNA double helix, means there is the separation of two strands of the DNA, it is said to be the formation of open promoter complex. After that, whenever there is the process of the initiation of mRNA chain synthesis, then it is considered to be the ternary complex, that is there is presence of RNA, DNA and the enzyme. If the process it is get stopped after the initiation of transcription, then it is said to be the abortive initiation. Abortive initiation after the clearance of the promoter. After the clearance of promoter, whenever means some nucleotides these are get added within the mRNA chain, then there is the continuation of this process of transcription. The first ribonucleotide it become unpaired, the polymerase then it loses the sigma factor. After the promoter clearance, there is the release of the sigma factor. Now this process of the transcription it is only catalyzed by the core RNA polymerase enzyme. Then in some cases it may be associated by the NUSA protein, which helps for the elongation of transcription process. That is, there is the addition of ribonucleotides according to the nucleotide sequences present on the template DNA. These are added to the 3' end of the mRNA chain. Here you can observe within that figure how there is the process of the recognition and binding of the RNA polymerase. Firstly, the holoenzyme, that is the core RNA polymerase and the sigma factor, they are recognizing the promoter sequences on the DNA double helix and they are bind with that. After that, there is the formation of closed or the promoter binary complex, closed promoter binary complex. Then whenever there is the melting of the DNA double helix due to the helicase activity of the RNA polymerase, it converts into the open binary complex. After that, the initiation of chain synthesis, there is the formation of ternary complex, and then whenever it passes from the promoter sequences, it is said to be the promoter clearance. Now this promoter clearance and the process of elongation of transcription, it occurs after whenever there is the addition of 4 to 10 nucleotides within the mRNA chain. The first ribonucleotides, they become unpaired from the antisense strand, that is from the DNA strand. The DNA strands reannul. Then the polymerase loses the sigma factor. After the promoter clearance, there is the release of the sigma factor and there is the recycling of the sigma factor. Due to the release of the sigma factor, now the polymerase enzyme it converts into the closed hand structure. Whenever there is the binding of sigma factor, it is an open hand structure. Now due to the conversion of this open hand structure into the closed hand structure, it gets surrounded to the DNA strand. Now the NUSA protein or the NUSA protein, it binds to the core RNA polymerase enzyme and it helps for the elongation of the mRNA chain synthesis, elongation of mRNA chain synthesis. And this RNA polymerase and NUS-A complex, it stays on until the termination process. The catalytic rate for the RNA polymerase enzyme, it is 20 to 50 ribonucleotides per second. There is addition of up to 20 to 50 ribonucleotides per minute within the mRNA chain. Here within this figure you can observe about the structure of the promoter and how the process of the transcription it can be carried out. That is, here there is the presence of the promoter sequences data box. The strand on which is acts as a template strand, it is called as the sense strand, and another it is said to be the non-template or the anti-sense strand. Now this anti-sense strand, it must be present within 3' to 5' direction, because as the RNA synthesis it only initiate within 5' to 3' direction. Then with the help of sigma factor, there is the recognition of the promoter sequences and binding of RNA polymerase enzyme. After the promoter clearance, there is the release of sigma factor and the NUS-A protein, it binds with it, and there is the elongation process will be continued. Now we are switch over to the last step within the prokaryotic transcription process, that is how there is the termination of this mRNA chain synthesis. Now this termination, it is occurs at the specific sites on the template strand which are called as the terminators. Now there are the two ways through which there is the process of termination of transcription it can be carried out. That is by the means of presence of the intrinsic terminators within the DNA sequences, or it is an row dependent terminators. So within the intrinsic termination, the DNA template on which there is the synthesis of mRNA chain it is carried out, it contains a inverted repeat sequences which are the G series sequences, and due to the copying of these inverted repeat sequences within the RNA chain, there is the occurrence of hairpin structure within the mRNA strand, that is, and then these are followed by 6 to 8 adenine nucleotides which are get copied within the mRNA as an uracil residues, because within the RNA instead of thymine there is the presence of uracil nucleotides, means the consequences of the poly U and poly A stretch. Here within this figure you can observe this inverted repeat sequence it is present, and due to the copying of this inverted repeat sequence within the mRNA chain, now there is the presence of this hairpin structure. Within that hairpin structure or the stem and loop structure, the stem it is an G series sequence, and again within that this stem it is followed by a polyuracil tail. Due to this polyuracil tail, majorly there is the release of the RNA polymerase enzyme from the DNA strand, and there is the release of the mRNA chain from this template strand, and also from the RNA polymerase enzyme, and through that there is the termination of transcription process. Now the another mechanism, it is with the help of an specific factor, that is the row dependent terminators, that is the row factor, which is an ATP dependent helicase enzyme responsible for terminating the process of transcription. Now it catalyzes the unwinding of RNA DNA hybrid, means as during the process of transcription this RNA DNA hybrid these are occurs, there is unwinding of this RNA DNA hybrid, it is carried out by the activity of row factor. It is an ATP dependent helicase enzyme. It catalyzes this unwinding of RNA DNA hybrid. Its rate it is 50 to 90 nucleotides per second. Here it is the mechanism of row protein or the row factor. Firstly, the row factor it binds to the transcript, that is the mRNA chain, at a row loading site upstream of the terminator, that is before the terminator sequences. There is the occurrence of hairpin structure it forms, and the polymerase enzyme, that is the RNA polymerase enzyme, it stalls at that site. That is whenever there is the hairpin structure it is get occur, at that time the polymerase enzyme it stalls at that site, and due to the helicase activity of this row factor it releases the transcript from the DNA template, and there is the termination of transcription process. there is termination of transcription process. Here there is the all over process of the transcription, how there is the initiation, elongation and the termination of transcription process. So within this lecture we are discussing in detail about how this process of the transcription it is carried out, what are the functions of RNA polymerase enzyme, how the initiation, elongation and the termination of transcription process it can be carried out within the prokaryotic cells. So if you like this video, please subscribe my channel, also share this video with your friends, and thanks for watching my video. Thank you.